if we have a unknown DNA sequence, then the array chip can provide the information of all the substrings that is present in that DNA fragment of size L. But it does not provide the information about the location of that sequences within those fragments. So if we have a sequence of string S of length n, the L-mer composition is the multiset of n minus L plus 1. Mers in S and is written as the spectrum as a SL. So we can see that if L is equal to 3 and S is the sequence that is TAT, GG, TGC, then the spectrum SL, it would be TAT, ATG and so on. How we can get this? Because this is the shortest information, we can say that this S is a superstring, and we can get the information from this superstring, and we will take the first 3 letter TAT, that is TAT, then we will move to the 1 alphabet on the right side and we'll start, instead of T we will start from ATG, that is ATG, and then we will start from the 3rd alphabet nucleotide, that is T, and we will say this is TGG, that is TGG, and so on. We can get GTT and we will reach up to the TGC. So this is the spectrum SL we will obtain from this superstring. So we can now formulate the problem of sequencing, that is the target DNA fragment from its DNA array data. So if we use this spectrum SL, we can also write these spectrum alphabets in the lexicographic order. That means we will write it in the alphabetical order because it is starting from A. So this ATG will come in the first position. Then after A, this is G. This will come in the second position. Then this is again G. Then we will see the second alphabet. If it is G and it is T, then it will come afterwards, and so on. So we can write it also in the lexicographic order. So we can solve the problem of the sequencing by hybridization. For this, we have the problem statement that we have to reconstruct a string from its L-mer composition. As we have seen in the previous slide, we have construct a string from its L-mer composition, and we have to input to the algorithm, and the input will be a set that is represented by S, and that S representing all the L-mers from an unknown string S. So the output will be, we will get the string S, this one string S, such that the spectrum SL, that will be equal to the S from the that was obtained from a set that represents the L-mers. So by using this, we can reconstruct a string from the L-mer composition.